have before me some lilies. One wonderful activity that you can do at home or at school is to dissect these types of flowers, especially in a reproduction unit. Lilies work the best. There's a couple of others that you could probably get your hands on. Daffodils, alstroemeria, tulips, and gladiolas also work well. Some differences in these sepals in their different levels of development. So you see very young in the flower's development, the sepals smaller and green, and you see it starts to elongate and turn yellow as it gets a little bit more mature. There's a mature flower. And here you see the sepal just beginning to open. It's elongated quite a bit and it's turned to yellow. And you can see the 3 sepals on the outside and the 3 petals inside, and you can even see some of the stamen and maybe the pistil a little bit on the inside. Here are the sepals, which protect the bud, the petals, which attract the pollinators. There's some pollen grains on one of the petals there. The flowers are angiosperms, and many of them are hermaphrodites. So they have male and female reproductive structures. Lily had 6 stamen, male reproductive structures, and here's 2 examples of them. So the whole thing is the stamen. The top part is the anther, which has the pollen grains in it, and this is the filament. Okay, the stalk here, and usually they stand up high above the female reproductive part, which is the pistil. All right, and this is the upper part of the pistil, is the stigma, which catches the pollen grains, and then we have the style. And I've cut open the ovary with a microscope. You could examine the ovules inside the seeds. This is the receptacle which holds the flower to the stem.